Welcome, everyone, and thank you for joining us. I'm Brenda Dettinger from BIOPAC, and I will be your moderator on today's webinar, Unlocking the Potential of Eye Tracking in Physiology Research. Today's webinar is being recorded, and we'll send each of you a link once the recording is processed. During the webinar, if you have questions, please submit them through the GoToWebinar control panel. That's how you communicate with me, your moderator, and I'll pass as many questions on as I can. Before we get to our presenters, I do have an announcement. We host a biennial human physiology training event, and registration is now open. This event will take place again in July of 2025. So we just passed July 2024, so I don't have to refer to that anymore, but definitely in 2025, it will be at UC Santa Barbara in California, and we'd love to have you join us. It's a great opportunity to get your hands on the equipment that BIOPAC offers and get some hands-on experience with all of the devices that we offer, including the devices on today's webinar. Here's just a few of my favorite photos from the 2023 event. So save the date, July 14th, 2025, and meet us in California. Now on to our presenters. I'm excited because we have two great speakers today. We have Dr. Marissa Biondi. She has a PhD in psychological and brain sciences from Texas A&M University. She's used her and has used FNIRS and eye tracking to study the functional organization of the developing human brain. She is currently the Senior Scientific Advisor for Tobii, and she offers funding support services to the North American eye tracking community. Welcome, Dr. Biondi.
Thank you.
We also have Fraser Finlay, the CEO of BIOPAC. He has over 25 years of experience in the life sciences and physiology monitoring industry. Fraser oversees the development of physiology data collection and analysis products and is a well-regarded expert who presents at workshops all over the world. Welcome, Fraser.
Morning, everyone. Welcome, Brenda. Thank you.
All right, Fraser, let's start with you. I'm making you presenter.
Perfect. Okay. All right. Well, Brenda, thank you for that great introduction. Welcome, everyone, to our webinar on screen-based eye tracking and the integration between Tobii and BIOPAC. But before we start, I've got a little background information about BIOPAC. BIOPAC develops high-quality scientific tools that allow you to measure physiology anytime, anywhere, with any subject. We've been in business for over 30 years, and 99 of the top 100 universities use BIOPAC products. And many of the largest technology companies in the world use our products for development and validation. And all those customers have now generated over 53,000 citations. So we're delighted to partner with Tobii and bring together two leaders in their respective fields. Tobii with their incredible range of remote screen-based eye trackers, and BIOPAC with its recognized position as the premier choice for physiological monitoring systems. The integration creates a seamless solution for recording physiological data, stimulus presentation, eye tracking, and video in one user-friendly solution. With powerful visualization tools and unique analysis options, the integration will provide researchers with new opportunities and streamline their workflows. To provide you with a taste of what we'll be seeing today, I have a quick glimpse of the new integration. Okay, so this is what we're going to be talking about today. For those of you that are familiar with AcqKnowledge, on the left-hand side, you can see the typical AcqKnowledge graph window.
Fraser, we can't see the AcqKnowledge screen. Sorry about that. Sorry for the interruption. There we go. Yep, now I see it.
Actually, give me a second. Okay, I'll have to stay. Sorry. Get these organized. Alrighty, so on the left-hand side, you can see the typical graph window that is AcqKnowledge. So we've got some physiological data in here, pulse, electrodermal, so this is skin conductance level. We've got which is this signal here is the tonic waveform. We've got the phasic waveform, the blue signal, second channel. Pulse from a PPG sensor recorded at the finger. Pulse rate. And then we've got some eye tracking signals down below. Over on the right-hand side, we have the visualization viewer that is a key component of the eye tracking. And if I click anywhere in the physiological recording, it will advance the visualization viewer to show the stimuli and the eye tracking response from the subject. So this was the image that was presented at this particular moment in time, and we can see part of the gaze path that was collected from the Tobii eye tracker. And then you can just hit play and play this back. So now this is just a replay of what was taking place while the participant was being presented with the different stimuli. So without getting lost in all the details at this point, I'm going to hand you back over to Brenda for our first poll of the day.
Great, thanks, Fraser. I'll go ahead and launch this poll. And we just want to hear from you about your experience or interest in using eye tracking. And we are going to take a question, excuse me, from the audience, from the registration process. So what is the distance? Well, maybe we should save that question for the next poll, because I think Dr. Biondi can answer that one. So let's just go ahead and answer this poll question. Then I'll remind everybody that you can ask us questions through the control panel on the GoToWebinar tool. So if you do have any questions for our speakers, please do pass them on. And I'm looking at the answers right now. Just another 30 seconds or so while people are still bringing in submitting their answers. Looks like the majority of people are currently using screen-based eye tracking in the lab. I would say close to 60% right now. And then about 20% are using glasses outside of the lab. And then 20 to 30% of people are looking forward to trying screen-based eye tracking glasses, and also eye tracking with virtual reality and augmented reality. Okay, well, thank you everyone for participating in our poll. We are going to switch gears, and Dr. Biondi is going to present more information about Tobii products.
Awesome. Thank
So
you so much,
Dr.
Brenda.
Biondi, I'm making you presenter. Yeah, there you go.
Great. Okay, share. Awesome. Well, thank you so much, Brenda and Fraser and BIOPAC, for inviting me to talk about eye tracking today. If you are new to eye tracking, I hope this answers some of the questions you might have and inspires you to add it to your own research toolkit. If you've used eye tracking, which seems like many of you have, then I hope you can figure out how to combine it with BIOPAC data streams, and then we can get to showcase some of your results here in the future. Here's our brief agenda for today. I'm going to go over briefly what is eye tracking and how Tobii systems use, why you might use eye tracking, and then showcase some eye tracking use cases and inspiration. And then I'll talk a little bit about our product portfolio, hardware and software, and then explain some of the use cases that might use one product or the other, so some of the differentiating factors in our hardware. So how do we see the world? This is an illustration of a scene that we might see in the world. It's just a shopping display in a grocery store. But the world doesn't really look like this to our brain. It looks more like this, where the only clear part of the scene is in the center and the rest is blurry. That's because the fovea is on the retina, and it contains the highest concentration of photoreceptors. So that means that your foveal vision is the clearest and crispest. It equates to about 1 to 2 visual degrees in the field of view, which is, if you hold out your thumb, it's about the distance, it's at arm's distance, it's about the size of your thumbnail. The number of cone cells decreases the further you get away from the foveal area, so the image gets blurrier. In the parafoveal area, it's still got some cone cells, but it's less acuity than the actual foveal area. And then everything else is in the periphery. So the rod cells are more prominent here. It picks up light, dark, movement, but things are really pretty blurry and monochrome. So when you move your eyes around a scene as you're looking, you are reallocating your foveal attention in order to gather information about the entire visual scene. Then your brain takes that information and melds it together into a coherent picture. But I'm talking about foveal attention because this is what is measured by eye tracking, not the periphery, but the exact foveal vision where you're looking and allocating your attention. So why would you want to use eye tracking and measure foveal attention? We think there are many compelling reasons that make eye tracking an attractive choice for your research, especially as it combines with physiological data. Eye tracking is objective and quantitative, so it allows us to capture data from participants without any subjective questions or conscious filtering or types of feedback that might bias the participant from answering. Eye tracking delivers unique insights into cognitive processes of your visual behavior and attention. So the eye-mind hypothesis is fundamental for eye tracking research, and this states that where you look at, in most cases, is what you think about. So being able to capture where someone is looking will tell you a lot about what's going on in their brain. Eye tracking is unobtrusive and non-invasive, so there are no chin rests needed for using our systems. They also plug right into a computer and blend in without biasing the participants. Eye tracking is versatile, portable, and easy to use, at least Tobii systems are. You can collect data anytime, anywhere, with systems small enough to fit into a backpack. And finally, eye tracking works really well with other types of physiological and neuroscientific measures, so GSR, heart rate variability, and all the things that you can, all data streams you can gather with BIOPAC systems to give you a holistic understanding of human behavior. So eye tracking is a great addition to data streams you may already be using. How do Tobii screen-based eye trackers work? There are 3 main components: cameras, illuminators, and algorithms. So the illuminators shine near-infrared light and project a pattern onto the eyes. We call these glints. The cameras then take images or pictures of the user's eyes and the glints. And then our algorithms have image processing models and machine learning algorithms to determine the eye position and the gaze position and the geometric configuration of those, to figure out exactly where someone is looking. Some of the common metrics and insights that you could get from eye tracking you'll see here. You could compare things like total fixation time across groups, or maybe even pre and post test. You can compare saccade length, so the distance between fixation A and fixation B, to look at, let's say, an expert versus a novice and how they scan a scene or try to do a task. You could even compare transition paths between conditions. So there's lots of different metrics depending on the types of questions that you're asking. So lots of dependent variables that you can use in your research studies. Eye tracking is used in a wide range of industries. So we have lots of eye tracking in healthcare, eye tracking in gaming, so being able to interact with video games to improve the mode of play. Eye tracking in extended reality. Many of you mentioned that you're interested in either VR, XR, or AR. We have eye tracking systems that have that are built into existing HMDs. Eye tracking is very common in education and training, and automotive. The automotive sector is growing quite rapidly. Driver monitoring systems are becoming very popular, and Tobii is involved in that as well by making sure that the person who's driving a car is actually paying attention. But what we're going to be talking about mostly today is behavioral studies and research. That's where we have been well established as the market leader. Our company's been around for 23 years, and we have solutions that fit all kinds of behavioral studies and traditional academic research. So here are some more examples. These are the domains where we have lots of customers. So most of you are maybe in psychology or neuroscience. This is an eye tracking and physiology webinar, or you might be in other kinds of related fields studying human behavior, but these are some of the main fields. Although we talk to people all the time from all sorts of areas of scientific research who are really trying to push the boundaries of science forward and using eye tracking in always new and innovative ways. We are proud and honored to have users of our eye tracking solutions from all over the world. As I mentioned, we are actually approaching our 23rd anniversary. So we started in 2001. We are in currently over 2,500 research institutions, including 99 of the top 100 ranked universities. It's come a long way. Our technology has changed and grown a lot. So that means that it's become more affordable, easier to use, more portable, and that way more fields can objectively apply eye tracking data to answer their research questions. So currently we're sitting at over 12,000 citations and growing over the last 23 years or so. But you can see on this graph that eye tracking publications have dramatically increased. So we hope that you consider joining our Tobii family and publishing some studies with our technology. Here are a few examples of some published research to get you inspired. So you can see these are actually heat maps that you're seeing on the screen. This is just one type of the qualitative data export you can get with Tobii systems, along with quantitative metrics and lots of other things. But you can see here, these heat maps really tell a story. On the far left, you can see attention to a face of a person with a mask versus without a mask, and that attention really does shift depending on if the person's wearing a mask or not. In the center, this research group studied coding behavior and the fixations that are included in understanding coding errors and fixing bugs. You can see exactly where someone is looking as they're scanning a block of code to find bugs. And on the very far right, the University of Bucharest in Romania used eye tracking in a classroom. So eye tracking can be used to teach students eye tracking methodology even as they're in undergraduate programs, and use it in their own marketing and consumer research projects to learn hands-on while they're still in school. So again, we look forward to reading your published work in the future if you decide to buy a Tobii product. So I'm going to talk about now our scientific research solutions. We have screen-based eye trackers supported by our Tobii Pro Lab software and our Tobii Pro SDK. We also have our wearable Tobii Pro Glasses 3 eye tracker, and that is supported by Tobii Pro Lab and our Glasses 3 API. We even offer various solutions together with partners, including the AcqKnowledge integration, which occurs via our Tobii Pro SDK. Our guiding principles for our products are tools that are valuable for you, give you good research practice, and deliver high quality results. And our products are only a part of the solutions that we bring. They're also supported by validation tests. So we have metrics reports in the lab and out in the field for our screen-based trackers and our wearable eye tracker, Tobii Pro Glasses 3, with large samples of population. So you can see what types of data you would actually be able to get in a real-life setting. We also have white papers, learn articles, materials, and overall support philosophy that we are here to be a research partner from start to finish. So that's what guides us in our relationships with our clients. So we believe it should be possible for everyone to conduct and analyze high-quality screen-based eye tracking and wearable eye tracking studies. So we designed Tobii Pro Lab to be an easy design and quick-to-learn software. It's a drag-and-drop interface, and it's an all-in-one platform for design, recording, and analysis. So some of the design features: easy drag-and-drop interface for images, videos, text, web, or even screen recording. We have advanced options for sampling, randomization, groups, and hierarchies. Advanced screen project for easy design options. So if you're testing maybe even hundreds of samples with a alternative force choice or visual world paradigm, Tobii Pro Lab can support that. And we also have easy co-registrations. Tobii Pro Lab has a TTL sync out 8-bit via toggle switch. It's included at no extra cost. You just click it on, make sure that your hardware is connected to your other physiological data acquisition tools, and Tobii Pro Lab will send out the TTL markers at the beginning of every stimulus, making your synchronization and co-registration very easy. For the record features, you can capture participant gaze, camera, and audio, and everything is very easily, the calibration is also here in the record feature, so everything is easily set up. And then for analysis features, you can replay the video recordings and inspect. You can draw AOIs, so those are areas of interest, physical areas on the screen that you might be interested in calculating the amount of fixations that occurred. Times of interest, so certain segments of the recording video that you want to analyze separately, maybe from an entire recording. You can view and export qualitative visualizations like gaze plots and heat maps that you've already seen. We also have all sorts of eye tracking metrics, some of which I already explained, and you can export all of them. Time to first fixation, total looking time, saccade, amplitude, duration, anything else you might be interested in. You can export all of these in a variety of pre-configured formats. Okay, so now we're going to talk a little bit more about our hardware. So this is the Tobii Pro Spectrum. This is designed to ensure the highest data quality and reproducibility. We have evolving patented eye tracking algorithms, sophisticated hardware design, and premium components. The Spectrum has a sampling rate of up to 1200 Hertz, and I'll explain a little bit more about the differences in the sampling rates in a few slides. But this will work with any kind of research, including microsaccade research or EEG co-registration. It also comes in 300 Hertz and 600 Hertz models. It is a dual camera system, so dual synchronized eye tracking cameras lead to higher quality, a more complete gaze data stream. There's a very large and tolerant head box, so you set the person in the middle of the screen and they can move around without any chin rest or head rest needed. So it is true stereoscopic measurement for locating the eyes of a free-moving user as they are looking at an XY plane of the computer monitor. The Tobii Pro Spectrum has bright and dark pupil illumination for increased trackability regardless of age, ethnicity, or eye features. And it optimizes between the dark and bright pupil imaging mode depending on the participant's features. Eye images are recorded and available for inspection, and it has a TTL sync in port. So where our Pro Lab software has TTL out and 8-bit, Pro Spectrum has a sync in port 8-bit for TTL. So if you want the Pro Spectrum to receive those TTL signals, this would be the right system for you. So the Pro Spectrum is our best performing system ever for extensive research into human behavior and eye movements. Next, we have the Tobii Pro Fusion, and I have one here so I can show it to you. It is a very slim, lightweight eye tracking system with a USB plug at the end. So when we develop this product, we want a high-performing but also portable system that retains high accuracy and precision when participants move or conditions change. So this is what we designed. We've replicated many of the technical innovations from the Pro Spectrum, but we've made it portable. So in short, the Fusion has a sampling rate of up to 250 Hertz. It also comes in a 60 Hertz and 120 Hertz model. It's also dual camera. It's also bright and dark pupil illumination for, again, that increased trackability. This will work with natively with 24-inch screens. It has embedded data processing. So all of the processing of the eye tracking data is actually here in our patented Tobii eye chips that are on board. So this doesn't use any extra CPU processing power from your computer system. So you have stable, reliable processing of eye images to calculate gaze and fixation. So this is a great portable solution for advanced research requiring high quality and performance. And finally, in our screen-based portfolio, we have the Tobii Pro Spark. This is our newest eye tracker. Here it is. You can see it looks pretty similar in the form factor to the Fusion. Here's the Fusion, here's the Spark. It's a little bit smaller. This is, again, our newest offering. It's meant to be an affordable entry-level system to get anybody up and started with simple fixation-based studies. So it is a 60 camera system, 60 Hertz system fixed. It is a single camera versus a dual camera. It does have bright and dark pupil tracking, embedded data processing as well. This works natively with up to 27-inch monitors. You can see it's very lightweight and portable. And this also works with our mobile testing accessory. So if you're interested in testing with either phones or tablets, and you want to use the mobile testing accessory and not our wearable glasses 3, you could use the Spark with that. So it's a small but powerful eye tracker, perfect for entry-level fixation studies. So often prospective customers will ask which system is the right system for me, or maybe they'll even use their budget as a guide, but we suggest that you consider other things, like what's your population? Are you testing adults, infants, children? Are you testing people with vision disorders, or maybe everyone across the age spectrum? Where are you going to be testing? Are you going to be fixed in a lab only, or are you going to be traveling with the eye tracker? And then what are your metrics of interest? Sometimes people don't know, so we can use this slide as a guide for what's possible. So if you see here that 60 hertz, 30 to 60 hertz is really good for fixation-based studies, just measuring how long is someone looking at X area on the screen. You can use any of our eye trackers for that. If you're interested in saccade, so how do people move their eyes from fixation point to fixation point? Maybe what's the distance between those saccades? How fast do they move as they're searching a scene? Then we would recommend our Tobii Pro Fusion 250 hertz or one of our Pro Spectrum, because you want to be able to capture those saccadic eye movements, which starts around 250 hertz. If you're studying microsaccade, so very fine eye movements, you need to use the Tobii Pro Spectrum 600 or 1200. The 1200 would be the preferred, but you can also use the 600 hertz model. So that's just one example of if you are interested in specific eye movements. But for other examples, if you are studying infants in a lab only and not traveling around without knowing much else, I'd probably recommend the Pro Spectrum. If you were studying children's learning and then going to visit them in schools, I'd probably recommend the Fusion. Or if you were using fixations to understand visual attention and having your students use the eye tracker to do some assignments in class, I might recommend the Spark. If you have more questions or have a unique use case, you can talk to us or the team at BIOPAC, and we can help find and recommend the right tool for you. Here are a few more examples from real published studies to help you understand some of the differences between the systems. So on the left, in a recent study, well, all of these were published this year in 2024, the Tobii Pro Spark was used to measure fixations in short-form video content. The Tobii Pro Fusion was used to understand eye dominance on fixation stability in school-aged children. And the Tobii Pro Spectrum was used to measure eye movements in Parkinson's disease. And they actually also co-registered the Tobii Pro Spectrum 1200 hertz data with EEG. So that was a really good choice for co-registering that high-end EEG data. These are real studies, so thank you so much. Back to Brenda for our second poll.
And thank you so much, Dr. Biondi. I'm going to go ahead and launch the second poll. And then while that poll, I am going to go back to that question that I had from the audience to ask you this during the poll. What is the distance the subject has to be from the eye tracker?
Yeah, that's a great question. So they all vary slightly, but in general, it's about 50 to 80 centimeters. So the specifics of each eye tracker, it may be 55 to 85, and you can see that in the they were on the web page in the specifics or the user manual for that eye tracker. But I usually say around 60 to 65 centimeters is a great place to put your participant, because that way they can lean forward and still be in track zone, they can lean back and still be in the track zone, and they can lean left and right, sort of optimize their comfort and movability while they're still being tracked accurately.
Okay, great. Well, thank you very much. So it looks like we are heavily geared towards neuroscience, psychology, and clinical research in this audience here today, with a real mix of every other application area, a little heavier on the human factors and engineering and driving and simulators area, but it's kind of like a decent spread amongst the other application areas. So thank you all for participating in the poll, and thank you, Dr. Biondi, again for all the work that you put together to share with us the information that you shared today. Now, Fraser, I'm going to make you presenter. Here we go. Switching gears.
Thanks, Brenda. Perfect. Okay. Well, that was some great information there from Marissa regarding the actual hardware and the Tobii products. Let's take a look at it from BIOPAC's perspective. And I'm going to sort of back into the live demonstration by showing you what and how the visualizations work and what you get after you've used the system. So I started off by showing you this display. So just to recap, on the left-hand side, this is a typical AcqKnowledge graph window, and you're seeing a combination of both physiological signals and the eye tracking data as well. So we've got gaze, X and Y, and there's a whole slew of these signals that can be brought in. So you can mix and match there. If we look along the top, just above the graph window, again, for those of you that are familiar with AcqKnowledge, you should be familiar with these, but these little light bulbs, these are indications that stimuli were presented to the participant. One of the nice things about the integration that we've got is the BIOPAC stimulus presentation element will automatically drop those event marks into the AcqKnowledge graph for you. So you don't have to worry about setting event marks up or anything like that. It just happens as a byproduct of the system. And whatever name you give your discrete stimuli, they will appear as part of the marker that is presented to AcqKnowledge. And then we also have things like hits on area of interest. So entering an area of interest, exiting an area of interest, and all those are marked throughout the recording. So you have a rich list of event mark styles. You can see the hits for the AOIs, you can see the exits, when stimuli are presented, etc., etc. So, and you can jump to any of these from this window. So this is taking us to the fixation, and you see we've jumped to that image. And then within the software, the cursor is aligned to that precise point. And that's the synchronization. That's the important part. Wherever I place my cursor, it will advance the viewer to that corresponding time point. Now, over on the right-hand side, the viewer itself, this is where once you've collected data from a participant, you get to see what the participant was looking at, okay, the image or video or PDF, web page, whatever it was that was being presented. You get to see that, and then laid over the top, you can select a whole array of different visualizations. So this one here is the gaze path, and we've got this toolbar along the top, and each time we select one of these, it creates a new view down below, and I'll get into those views in a little more detail. In AcqKnowledge, we've got the same toolbar available above the graph window, so you can jump to these very quickly just from either within AcqKnowledge, or if you're spending most of your time over here in the visualization view, you can do it up top here. So just sort of, it's a nice tool, nice feature that sort of makes it a little bit easier to navigate your way through. Then again, in the visualization viewer, you can drop into any of these different stimuli that were presented. Okay, so it's just a list of each of the stimuli. This is a beach scene that was presented. But one of the nice things about the visualization and gaze path, it's like a video recorder. So if I hit the play button, the system will now play the movie back that was recorded during the experiment. So here we can see precisely where the participant was looking. You see their gaze path, and just plays it back very, very nicely. And you can do that just manually. If I click this point in the image, as I advance the cursor across, we can see precisely what's going on at that specific moment in time. Okay. Now, I mentioned this toolbar, and I've already shown you the heat map. So very quickly, you can get to a heat map, and you've got controls for all of these different tools. So if I hit this icon here, the little slider bars, you can make some adjustments. Now, the way the heat map defaults, if you select an image, it will give you the complete heat map for the image, but you can also show this as a rendered display. So as I highlight an area of data, you can see that heat map growing as well. Okay, so you can do it one of two ways. And each of the different visualizations has different controls. Now the pie chart is based on the areas of interest, and I'll get into that when we move further along. But basically, on any image that is presented or any stimuli that are presented to the participant, you can select regions as specific areas of interest. And with those areas of interest, you get particular pieces of information. So in this case, this was the sequence of shapes and different colors. So this was the square that was presented. We can see that the time on the square was 4,678 milliseconds, or a little over 4.5 seconds total time. The person was actually looking at the center of the screen because of the fixation that occurred before. So immediately the image was presented, the first hit was on the square because that was the center image. There were 12 fixations while the participant was looking at the square. And then we provide some extra pieces of information, and these can be from any of the physiological signals that were being presented. So here we're showing the pulse rate, the average pulse rate while the person looked at that part of the image, that area of interest, was 49 beats per minute. So over that approximately 4.5 seconds, the total average was 49 beats per minute. And then in terms of the electrodermal activity, we're looking at the skin conductance level here. So this is the tonic waveform. You can see that the average was 4.99 microsiemen over that same time period. And then when you jump into these different areas, this is the red circle, and you can see the heart rate is at 51, and the average EDA, skin conductance levels, up to 5, and then down to yellow. So slight differences, not a very scientific study, but it gives you an idea of how those work. If we come into this view here, this shows us the actual areas of interest that were placed over the elements. And just above the graph bar, we've got this sigma icon. If I select that, these are the measurements that I was talking about. And I can come in and select additional measures for these. But at the moment, I've got in there microsiemens and rate. So it's easy to add those. And then once we've got those measures, and we'll come into this in a moment, we can export those in our report. Now, just some different views to give you some other ideas. This is a luminance. Actually, maybe let's just change an image to maybe a little bit more compelling. Here we go. So we can see where the bulk of the time was spent. It's kind of a little similar to a heat map. So you can see a lot of the area was spent around here. The luminance just kind of highlights the primary area of activity. This view here gives us an indication of the areas of interest. So let's pick another image where maybe we've got some more areas of interest. Here we go. So here we've got the bug, the right eye, left eye is green, and you can see the amount of time the person spent just as this scarf plot. And then finally here, this just gives us the sequence of the areas of interest. So C, A, B, C, C. This was the sequence that the person looked at. The time is not relevant. This is just the pathway based on the areas of interest. All right, so then you get a very rich report that comes from the system. If we select this option up here, create the area of interest, I'll include the fixations. We'll run the report. And this will open up in Excel. It takes a couple of seconds to run. All right, so now Excel will open. Completed the report. All righty, let's bring that across. So now we've got each of the images that were presented. So these were the shapes: red circle, yellow circle, square. We get the dwell time, the number of entries to that area of interest, the time of first entry, minimum dwell time, maximum dwell time, mean dwell time, the number of fixations, the time of first fixation, the total fixation time, standard deviation, EDA, and a heart rate. And then that's the same information available for each of the images based on the number of areas of interest that were added to that particular image. And then we get information about, let's back up a little bit. So this is the sequence, the dwelling sequence. So you can see the path that the person took and the dwell time over those particular areas of interest. And then down at the bottom, we'll have a fixation report. And these are the fixation sequences. We can get the coordinates and the fixation time, and then the name of the image that was presented. And that's the same for each of the slides. So very rich report. And then as far as the physiological data is concerned, any of our analysis here, I've already analyzed this. This is the skin conductance level again. Come into the analysis menu. You can come in and select electrodermal activity. You can locate the skin conductance responses, derive phasic, etc., etc. And then the system will add these event marks that show the skin conductance response. The first parenthesis shows the onset and the second shows the end. And then you can take any measures you want from these and export those as well. So very easy to get measurements from the data and combine with your eye tracking in one user-friendly interface. So I'm going to hand you back over to Brenda, and then we'll go through a live demonstration and show you how the system comes together from a recording perspective. So Brenda, I'm going to hand back over to you.
Oh, we did have a question about like a larger monitor. Maybe this is a question for Dr. Biondi actually, and so maybe we can address that later. But a couple people are asking about how it syncs the data. I don't know if you want to address that now, or if you want to just cover that in your presentation.
I can answer that now. So everything is fully integrated. There's an SDK that comes with the Tobii eye tracker, which is what Marissa talked about before. We take those data streams and we make sure that everything is synchronized within AcqKnowledge.
Okay. All right. Well, thank you very much. Just I'll go ahead and end this poll here and let you know that, let's see, it was heavy on the ECG, EMG, EEG, EOG, and EGG metrics, and EDA. Those were the top two responses. And then respiration, pulse, and apneas was kind of like equally shared, with apneas being a little bit lower actually than pulse and respiration. So I'll go ahead and close that poll. And then Fraser, I know we're going to move to another computer for you, and I'm going to make you presenter on that other computer.
Thank you, Brenda.
Okay, great.
All righty. So good. Let's get a little bit organized. So I thought the easiest way to show this is from the perspective of a live demonstration and give everyone an idea. So with the BIOPAC stimulus presentation, you can open up the stimulus presentations from the main graph window, and this is what the user interface looks like. So this is completely embedded within AcqKnowledge. There are three tabs: stimuli, sequence, and data recording. Stimuli are basically anything that you want to present to the participant. Now, I've already created this one. If I click on any of these, I can change the name of it, show the image on the right that's being presented, and I'll come back to random in a moment. Let's back up a little bit. So I've got an image here. On the right-hand side, I've got the properties. So it displays what's being presented. And then on the right, I have some controls. The controls cover the scaling of the image, the way it's being presented. If the image doesn't 100% fit the screen, you can control the background color. You can control the time that the image is presented for. And then by default, something I mentioned before, the stimulus delivery event is presented, but you can choose a different type of event mark. Just depends on what you're doing. The background color is controlled. Scaling of the images, just simple checkbox. So this is, the original size of the image. Obviously, it's not going to fit my screen. I can elect to maintain the actual aspect ratio of the image once it's rescaled. So now the image will be presented the way I want to show it to the participant. Now, to add different types of stimuli, we've got a plus, and then you can pick the type of stimulus that you're going to add. So for the purposes of this demonstration, is going to be predominantly image-based and a little bit of text, just to give you a flavor for what we can do. But very easy. You select what you want to add, and then you can come into your folder of images, and then you can just grab any particular image that you want, and it will drop it down into your list. So that's how we add the stimuli. The next part is one other thing that I should mention. You can also categorize your images. So let's say you've got a study where you're presenting positive and negative types of stimuli. One thing that can be really helpful is if you put all your negative images in one category. So here, this particular image here, I've got it labeled as negative. It's very easy. We've already got, positive, negative, neutral. You can manage those categories. You can create your own categories, or you can just select none. The advantage of that is once I've got a whole bunch of negative images, I can just have the software randomly select them. So I can create a randomized collection of negative images. So this is just going to pull any image that is available with a negative property. Now that is all managed through the sequence. So here we can control precisely when the different images are presented. So in this case, a fixation cross appears, then we have one of our negative images, then a fixation, then shapes, and it runs through from top to bottom. Very easy. Then over on the right, these are the stimuli that appear in our list here. Okay. And just to move any of these across, you just select the image and then you can just move it over. So now we've added cockroach to the end here, and I'm just going to remove that. Okay, so pretty easy. Now the data recording, this part manages the data collection part of it. So we've got an MP160 connected. I'm actually wearing a bioinomatics transmitter with a pulse sensor and electrodermal activity. I can come in and edit that current setup, and you can add any module you want. So those of you that are familiar with AcqKnowledge, you've got this long list of devices that can be added. Pretty straightforward. You can control duration and etc. etc. But really, for the most part, this is all you have to do is set this part up. Enable the eye tracker. Then you've got this area of interest editor. So the area of interest editor takes your stimuli, okay, and it allows you to create the areas of interest around that, and it uses these tools along the top, and you just create the areas of interest that are relevant for your study. And then once you've saved those areas of interest are saved for your specific stimulus presentation. Along the top, we've got options for, you can do a dry run. So that means that you can test your stimulus presentation without recording any data. The red circle indicates that you want to record. So when I hit the record button, I'm going to say, yes, I want to save everything. I'm going to retry, and I'm just going to give this a new name. All right, so at this point, we're now able to start running our setup. For the purpose of this demonstration, I'm just going to use a 2-point calibration just to keep it a little bit short and sweet. This is the Tobii eye tracker that I'm using. The serial number is listed there. I can identify the monitor that is going to be used for my stimulus presentation. Now, typically, you have two monitors, one for the technician, usually a computer or a laptop, and then a second monitor that is used just for displaying the presentation. And then the technician sees all the data coming in and precisely where the participant is looking, but the participant is just seeing the stimulus presentation that you've designed. So I can identify that. It colors it green, so we know, okay, this is the display that is going to receive the stimulus presentation. When I hit play down below, I get an image of a head, and we can see the eyes. And all I'm going to do is hit ready. And now we'll go into our calibration. And all I'm doing is just looking at those red crosses in the green dots. We can see just a 2-point calibration. I'm not sure how well this is showing up for you, but there's 2 little hourglasses within the confines of this green circle. So I'm going to accept that and say the calibration is good. Now everything is going to start in 3 seconds. First, we've got our fixation cross, and now I'm seeing my first image. And just going to be looking at these images. Another fixation cross. These would be considered our negative images. A more pleasant image to look at. Now in this case, I didn't mention this before, but the last area, the last word in that sequence there, I created an area of interest to advance to the next stimuli. So the moment I hit I think the word is AOI, then the software will immediately jump onto whatever the next image is in the sequence. Okay, perfect. So now the experiment has ended. We've got the ability to go in and look at the visualizations and the data. So let's take a look. Okay, so perfect. Top channel is my pulse. The second channel is the EDA. And we can go through and take different types of measurements. We can run through the skin conductance analysis like I showed earlier. But before we do that, let's just make sure we've got some viable data. So again, I'm just opening up the gaze path display. If I click anywhere in the graph window with my cursor, it will advance to the corresponding image. And then I can hit the play button down in the bottom, the green arrow. And now we'll see the gaze path plotted over the image that was present at that moment in time. Okay, so that looks like it's worked. One thing I want to do, because I would like to add a couple of measurements, I'm going to calculate the pulse rate measurement. I didn't add that as a calculation channel, but we can add that after the fact. So this output, care about that, and actually select all data, and then we'll do that again. So this will just give us the rate calculation from the pulse data. Hit OK. OK, perfect. So now we've got this second channel here, and I'll use that in a moment when we come in and do the analysis. So again, I've got the heat map option, so we can see the heat map, and you can control all of these views. This particular display will give you the heat map for the entire recording period that the person was looking at that image. But you can also create a heat map based on specific time periods. So to do that, under the control option here, we can use what's called an animated heat map. And if I just highlight an area, let's find that image again. If I highlight an area like so, we can see as I move the cursor across, we see that heat map change. So the default is to provide the heat map for the entire period that that image was displayed for. But if you don't want to do that, you just maybe only care about the first 5 seconds or first 10 seconds, you can just select that region and the software will build the heat map for you. And then you can control the sensitivity here based on the colors that are being presented. Now, the luminance will give us a view of where the majority of the time was spent on that image. So in this case, we can see the majority of the time was spent on the side of the face, the mouth, obviously where the spider was, and over the one eye. The pie chart will provide us with information for the areas of interest that relate to that particular image. So the spider, the mouth, and the right eye. The spider, almost 7 seconds was spent looking at that. The first hit was 608 milliseconds in. 23 fixations whilst looking at the spider, then the right eye, and then the mouth. On the left-hand side, when you mouse over one of the segments of the pie, one of the slices, you get a measure for skin conductance level. So that is the average skin conductance level while the participant was looking at that specific area of interest. So in this instance, I spent almost 7 seconds looking at the spider, and that's not 7 seconds continuously, that's 7 seconds across the entire 10-second window the image was presented. So that means we've calculated the skin conductance level, the average of that, for all the time that I spent looking over it. And then the reason why I wanted to add in the rate calculation, we now have my heart rate at 50, almost 52 beats per minute while I was looking at the spider. Now to do that, there's a little sigma icon above the window. When I open that up, I have this floating bar here, and this is always visible, whereas the information on the left only becomes visible when you mouse over that area, whereas this will always be on screen. So here we can see while I was looking at the mouth, my heart rate was 56 beats per minute, and skin conductance level was 8.3 microsiemens. Right eye, almost 55 beats per minute. Spider, approximately 52 beats per minute, and we can see the skin conductance level changing. So really rich information that very quickly we can extract from this data. You can still use all the tools within AcqKnowledge to get all the other types of measurements that you're interested in, but here very quickly we're getting unique pieces of information that would take a considerable amount of time to determine. Okay, other options beyond that, we have the scarf plot. So the scarf plot gives us the path the person took as they passed over the areas of interest. You can add in the time the person was looking outside of the areas of interest, or you can just throw that in. So here we can see that this is the total period of 10 seconds. There's a small amount of time where I wasn't looking at any area of interest. These are broken up by the time that I spent looking at that area of interest. Okay. Excuse me. And then finally, this string plot just gives us the path of the areas of interest that were hit. And it gives them letters: A for the spider, C for the mouth, and B for the right eye. And this is the path ACBAAA. And when we extract all of this information into a handy little report, I'll just run that report. I'll include the fixation sequence as well. This will export all of that information into Excel. Here we go. If I just run through here, it gives us a summary. There's information for each of the images with each individual area of interest. So this was the shapes, red circle, yellow circle, the square, and then the background. So the background is always included. In this case, there was no time spent looking at the background. I was either hovering over the red circle. You can see the number of times I entered that area of interest, the time of entry, the minimum dwell time, the maximum dwell time, the mean dwell, the number of fixations, the time to first fixation, and then we get into the heart rate and skin conductance level values as well. If I scroll through the data, we then get to the AOI dwell type in milliseconds. And then at the very end, this is the fixation sequence report. So for the spider, the first time that was presented, we get the time of the fixation, we get the coordinates, and then we get the label for that. So this is background, this is the spider, and then this works through the entire time that image was presented. So you get all of the fixation image as well. So a lot of information, very quick to grab that using the report option. Okay, so that sort of allows us to look at the data that we just collected. Anyone that's familiar with AcqKnowledge will immediately feel at home with the user interface. I mentioned this before, but just as a reminder, these are the icons for the events that were presented to the participant. Useful to understand what was going on. If you're in the gaze view, you can, just by clicking anywhere, you will see the corresponding image advance. And then from there, you can create a gaze path plot. You can control that. I showed you how to make adjustments to the heat map. You can make the fixation points constant in color as opposed to raindrops. You can have, at the moment, the default is for the tail, that the longer the history of the path goes on, the fainter the older part of the data is. The newer data is the darkest. You can turn that off so it's consistent for all of them. You can increase the size of these. So sometimes if you're making an image or you're trying to show this to people, it can be a little bit easier if you increase that size. And then obviously when you play this back, probably a little bit easier on the eye. So let's take a look. You'll notice that these are all constant in size now. So then one last thing that I wanted to show, if I save all of this, I'm just going to save this, and we'll just call it, oops. Save that. Next time we open this up, it will open both the visualization viewer and that. But one thing I wanted to do was come in here and look at the area of interest. So this is that beach area, and actually the one I wanted to focus on is the shapes. So these are the shapes. I had 3 areas of interest created for them. I can just click on that area of interest. On the left-hand side, you can see it as red circle. Now this is the part that's super important. When a participant hits that area of interest, you can choose for certain things to happen. I had beep presented, although I actually had my volume turned off because it was creating some feedback. But when my eyes hit this area of interest, a beep would have been heard through the computer. But we could select to advance to the next stimulus, so advance to the next image. We can start the stimulator, so BIOPAC could deliver electrical stimulation to the participant. We can control third-party devices just based on the area of interest. So you can do that from an image or stimuli perspective. So you can deliver electric shock, for example, as a specific type of stimuli, or you can do it as conditional on the participant performing some task. So in this case, when the person looks at that particular image, one of these tasks will occur. Like I said, I had it set to beep. You can also do it on exit. So in the case of the stimulator, you could have it start the stimulator and stop the stimulator. So maybe you've got an olfactory stimulus there. In our case, we have a scent delivery system. We could enable the delivery of a scent and disable it when they turn their eyes away from that image. For the text, if I go back here, this is the text that was displayed. You can see at the very end, I've got an area of interest that contains, I created one area of interest around the AOI last word in the sentence. And I had that set up, if I click on that, so that it had advanced to the next stimulus. So the moment I reached the end of that line, the system immediately rolled into whatever the next stimulus to be presented. Anyway, that gives you an overview of the integration with the Tobii eye tracker. The one part that I didn't show, which I want to quickly, Tobii have this nice setup that allows you to customize the configuration for the display that you're using. So I've got this one set up already for my laptop, but if I create a new one, I'll walk you through the options here. You can determine whether it's what's considered a regular setup, which would be that the eye tracker is attached to the screen, or if it's a more advanced setup where the eye tracker is in front of the screen. So for situations where you've got a larger display, what I'm doing here is considered regular. So I select next. Oh, I didn't select. Let's do that again. There we go. So now I've selected the eye tracker that I'm using. I can then adjust the height of the actual eye tracker relative to the monitor. So for my particular monitor, the eye tracker overlays the bottom of the display just a little bit. So I can put in an offset there. I'll put in, I'm about, the top of the eye tracker is about 6 millimeters above the bottom of my display. And once I put that in, I can hit next. Oops. hit next. And then we've got these two bars here, horizontal bars, that allow me to adjust to precisely where there are specific markers on the top of the Tobii bar, white markers, and all I've done is align those to the display. Then I can hit next. Then I can create a new setup name. So maybe I'm using two monitors and I want to give this a new name. So, let's call this laptop 3, and then we save. So now I can come in and choose the precise monitor that I'm using. So pretty easy. It's kind of a nice little tool to get everything set up for you. Anyway, I'm going to hand over to Brenda. We've run a little bit long, Brenda, so hopefully we've still got some time for questions.
Yeah, I think we should ask some of these questions. I know we are a little bit long, but I think it'll be real helpful to have you guys answer some of these. So, all right. So let me just share a couple of things with you. I'm going to make myself presenter here. One sec before we get to the Q&A, because I am getting a lot of questions. So thank you, Tim, for jumping in and helping answer some of them and helping me prioritize what to ask. So, we're formally entering the Q&A portion of our webinar, and I just want to share that you can reach out to your local representative. We have representatives all over the world, and you can visit any page of our website and scroll down and click this contact button right here, and it'll send an email immediately to your local representative. Mine is Amy because I'm in California, but if you're in the UK, you'll see your rep for the UK and all over the world. We also do a bunch of webinars every year. Actually, we have over 100 webinars on our website. So if you wanted to visit any of our other webinars, you're more than welcome to. You can also rewatch this webinar. I know a lot of you have had to drop off early here. So we'll send a link to the recording of the webinar, but you can also just jump into this page on our website and rewatch. So let's go ahead. I'm going to just pop open this here, and then let me get into my questions again. All righty. Let's see. Okay. All right. Let's see here. So a few questions about the stimuli, Fraser. One of them is from Joanna. Are we able to add questions after the stimuli? And also, I don't know how to say this name. Lakarzar asked about adding HTML link as a stimuli as well.
Yeah, you can add web pages if I think that's what they're asking for, and then asking questions. Yeah, yeah, you can. It just depends on precisely what you're trying to do. That's kind of a little bit nuanced, but yes, you can display text, and there isn't a way to collect the yes, no. I suppose you could do it through the eye tracking. So if your participant is answering a yes, no type question, then they can look at that area of interest and it would collect the data that way. We also have the rating scales and dials that are available as well, so they can rate images or how they feel about something, which may also align with the question.
Okay. Okay. Well, if you have a follow-up question, let me know. And also you can reach out to your local representative. I just showed you how to do that, and then they will get the answers to these questions. Okay, so Valerie asked about the visualization viewer. What software is that in? Is it in AcqKnowledge or in Tobii or both? I know you were just showing AcqKnowledge, Fraser, so all of that was presented through AcqKnowledge. Did you want to say
Everything
more
was
about that?
presented through AcqKnowledge. Yeah.
Okay. And
Actually, Brenda, just to be clear, there is a license for that. It's a licensed feature. So when you buy an eye tracker, you get the license as well, and that includes the stimulus presentation and the visualization viewer.
Okay, great. And this question was from a long time ago from Greta, so thanks for hanging in there, Greta. I think this is probably for you, Dr. Biondi. I know there is no exact value, but low values for accuracy and precision are good. Is there a limit how high these components can be to say it's still a good validation or calibration?
Great. Yeah, that's a really good question. I brought that up briefly when I mentioned that our screen-based eye trackers have accuracy and metrics reports. So these metrics reports will give you an idea of what you can generally expect. You also might get precision accuracy numbers that are better. Ultimately, it's up to you what you might want to exclude from your participants. So fortunately with well, I don't know how it works in the AcqKnowledge software, but in our software, when you do the calibration, you can actually get the quantitative metric for that calibration for that participant. So you can recalibrate as needed, and then you can say, okay, well, this, later on, this participant's calibration score was too high. So I hope I'm answering your question. There isn't really an upper limit on the metric. It is really up to you, and just whatever you decide. You can look at the metrics for the hardware and say, zero point 0.3 visual degrees is sort of like a standard accuracy for, I think, the fusion. And if somebody is at maybe zero point 0.6 or zero point 0.7, you might say, well, that's sort of on the border. If they're at like 1.0, maybe you want to exclude that participant. It is really up to you. Just keep that keep those notes of the participants that you exclude and the rationale for your publications, so you can explain why you excluded or included people. Hope that answers your questions. If you have any more, let me know.
Okay, yeah, we'll organize all of these questions and we'll produce a Q&A document from because there was a lot of questions today, so we'll go ahead and get that organized for you all. And then another follow-up question for you, Marissa. This came in earlier. Can we use a larger monitor or have a person further away from the display screen?
So yes, you can use a larger monitor. When I gave the metrics for 24 inches for the Fusion, 27 inches for the Spark, those are in the standard configuration. So that means it's really easy to set up. The eye tracker goes directly on the monitor. The Spectrum comes with an integrated 24-inch monitor, but you can also remove that monitor and use it what we call a non-standard configuration. You can also use the Spark or the Fusion with a tripod in a non-standard configuration. The participant still needs to be within 50 to 80 centimeters from the eye tracker, but then you can move and shift the eye tracker and the participant further away if you have a much larger screen. It will require non-standard configuration to set up and some measurements and geometry, but all that's possible through the eye tracker manager tool that Fraser showed. So yes, larger monitors can work, but the participant still has to be within the same range from the eye tracker, but you can move everything further back as you need.
Okay, great, thank you. So one last question, and this is actually a compilation of a lot of people's questions related to AcqKnowledge and the integration. One of the questions specifically was about how are we able to collect data of pupil dilation with AcqKnowledge. That seems to be closely linked with the other physiological data. How do they integrate all of that data together? I mean, this is really a powerful tool that we just saw integrating all the physiology data. Is it unlimited number of single signals? Any type of physiology measures can be used in this? Just talk a little bit more about how AcqKnowledge integrates all of that data.
So just about any physiological signal that you can collect with the BIOPAC system can be combined with any of the signals that come from the Tobii eye tracker as well. So, you have a rich combination of options there. It just depends on what you're doing. I mean, there isn't a restriction other than someone that wants to do this in an MRI. None of this stuff is MR compatible. But aside from that, any of our regular physiological monitoring, whether it be a C series amplifier, a D series amplifier, wireless bioinomatics, that will all work with it. And you just pick the signal that you're interested in, and then you can pick the signals that are relevant from Tobii, and they just show up as different channels in the AcqKnowledge graph window.
I want to make a point about pupil diameter. So all of our Tobii eye trackers provide left and right eye pupil diameter metrics. That's included along with the other fixation metrics. If you are interested in using pupil diameter as a dependent variable, I would recommend not to use the Spark. The Spark is a single camera eye tracker, and we found that our single camera systems have more more issues with the Z distance movement. So the person is not restricted as they're sitting in front of the eye tracker, but if they move forward, the pupils in the data will actually look larger as an artifact of them being closer to the eye tracker. The dual camera eye trackers of the Fusion and the Spectrum handle this fine. They're able to, because it's stereoscopic measurements, they are not affected by moving closer to the eye tracker and showing an artificially larger pupil. So while all of our eye trackers do provide pupil diameter measurements, if you're going to be using that as an independent variable, or as a dependent variable, I would not recommend the Spark. I would recommend Fusion or Spectrum. Our glasses are also compatible for that too.
Okay, well, thank you both very much. I know we're way over, so I'm going to move on here. I just wanted to show you a little sampling of some of the webinars that we've hosted over the last several years. Again, you can reach out, you can join us at biopac.com/webinars to rewatch any of those recorded webinars. We also host workshops all over the world. So if you're interested in hosting a workshop in your region, we can come to you, we can bring the equipment to you, you can invite people from your institution, you can invite people from the regional institutions. We do this throughout the year, and we'd love to visit you in your location. You can just contact your local representative to learn more about that. And then before I give my closing comments, I just want to thank Dr. Biondi and Fraser for all the work they put into today's presentation. It was a very exciting webinar, and I really appreciate your time and the effort that you put into that. For the rest of the audience, today's webinar was recorded. We'll email you a link, any slides we have, and the Q&A document over these next couple weeks as we get things organized. When you close the webinar window, you'll see a survey. Please complete that so we have your feedback and ideas. Seeing lots of love and applause for the two of you. Thank you all for participating in that. You can also visit BioPAC. We have a ton of resources. I didn't show you that, but if you go to our support section, there's a lot of additional information, including screencasts on specific features, analysis tools, application notes, step-by-step instructions, more videos, and of course, information about upcoming events. We will be at Neuroscience in October in Chicago, so come by and see us there.
We will too.
Yep. So we'll see you there, Fraser and Dr. Biondi. It sounds like we'll be there as well. So come by and see us, and also we'll be at other events throughout the year. Anything else before we conclude, Dr. Biondi and Fraser?
Thank you so much for inviting us. It's really exciting to see how our two products work together, and thank you all for attending and great questions.
Thanks, Marissa. Thanks, everyone. Thanks, Brenda. Have a great rest of your day.
This concludes our webinar today. Thank you again, and have a great rest of your day, everyone. Take care.
Bye.
Bye.